wait until she is done taking photos. Hi everyone, my name is Erfan. My mentor is Ryan Lack, and together we're creating a new optogenetic tool to control kinase signaling in Wilson Lab. So we all have experience taking an intro biology class either in high school or college. So we might be familiar with the phrase central dogma. Central dogma is essentially DNA turning into RNA, and RNA turning into a protein. But what if I told you there's a world of hidden processes that has to take place in order to allow DNA to turn into RNA? One of the major components of these processes is a protein called kinase. For my project, we are interested to create a tool that is going to activate or deactivate the function of kinase by shining different light wavelengths of light to this tool. So you might be asking me right now, what do we even care to activate or deactivate the function of a kinase? Well, it allows us to have control over central dogma. So essentially, if there's a specific DNA we don't want it to turn into RNA, we can stop that process. Also, this tool is very precise, so it can be used in the cell of drugs, which tend to have off-target effects. So currently, without this tool, we're at the red light. We can't move forward to know about kinases more deeply. But with the creation of this tool, we'll be at the green light to fast forward and see what the actual specific of kinases are. So, what's the vision? Well, my vision is to have control over kinase named ERK. I know, it's just like my name, ERK. So, ERK is the last kinase responsible for allowing DNA to turn into RNA. Now you might be asking me again, what do you even care about controlling ERK? Well, think of a cell as a factory. In order for a factory to function properly, it needs to have recipe for each set of product that they produce. But sometimes we have lazy work in these factories to change a recipe for a new product when the old product is completed. In return, this results in excessive amounts of old product, which is a big no for a factory. And it's exactly the same case for cancer cells. The cell's recipe, DNA, is being read too much. So, in return, this results in excessive amounts of RNA, which is also a big no for the cancer cells. So one way to stop this is actually to deactivate the function of kinase when it enters ERK. By deactivating the function of this, we can stop the transformation of normal cells to cancer cells. So how does this tool even work? Well, we're going to modify the kinase, ERK, by attaching two light-sensitive proteins over the active site. So these light-sensitive proteins act like a gate. When we shine infrared light at these gates, the gate will open up and expose the active site. When the active site is exposed, the kinase is activated, so DNA can turn into RNA. But let's say we want these gates to close now. So we shine red light, and these gates will close up and block the active site. When the active site is blocked, DNA can no longer turn into RNA. What a magic. So, as you can see, I want to show you guys the actual steps we're going to take to make this tool. There are 4 crucial steps, so we'll go one by one and dissect each. First, designing a plasmid. Imagine all these arrows represent proteins. There are millions of proteins out there in the universe, so we have to find the right specific proteins to construct our plasmid. After doing some research with Ryan, we actually found perfect proteins to make our plasmid. But let me show you the real plasmid. This is cartooning. So, kinase of interest, ERK2, is attached to 2 light-sensitive proteins right here. But the question becomes, how do we attach proteins to each other? Well, this is done by cloning, which is our second step of the project. Cloning has few steps to itself, but we wouldn't want to go too deep into detail. One of the major reactions in cloning is called Gibson reaction. What it does essentially is you can attach proteins over their complementary sites. But unfortunately, Gibson is not always perfect. What happens is that these proteins may end up attaching somewhere else, or worse, don't even get attached at all. Just like this picture. As you can see, the proteins were attached somewhere else. That's why we didn't get this nice circular plasmid shape that we hoped. So how do we even know that we got the plasmid that we wanted? It's an easy step. We get the plasmid that we think we got, add it to some enzymes, which will cut at specific sites, and then let the magic happen in the tube. We're going to extract some of that reaction and load it on a gel. And this technique, as Ricardo mentioned earlier, is called gel electrophoresis. So technically, the bigger fragment stays on top and the smaller fragment stays at the bottom. It's just like carrying boxes. When you have too many boxes in your hand, it's heavy, so your travel distance will be very short. But when you have one box, it's lighter, so you have more energy, you travel farther. And so this was our case for the plasmid. The bigger portion, which is this, like the heavier portion, traveled short amounts. But the smaller one, since it's lighter, traveled the farthest. So, and after doing a lot of reactions of cloning and Gibson, and unfortunately we didn't get always the perfect results, but finally, about a week ago, we got the exciting news that this plasmid worked. So we were super stoked we can go to the next stage of the project. I know, y'all love bring it up. So, next step is actually inserting the plasmid, the one we just got, into the virus. Like the animation. The next one, we're gonna get that virus that we just got and inject the plasmid into the stem cell. And once we have the plasmid in the stem cells, we're going to use this fancy optogenetic microscope to control these plasmids in the cell. So this is where we can turn on and turn off ERK multiple times. We can do 1,000 times, however you want, until you get tired of it. So turn off, turn off, turn off. And that's when we can actually get real data to see if turning on or off of ERK works. So to recap, we're currently at this stage of inserting the plasmid to a virus. Hopefully we'll get that done in the next couple of days, and then we can proceed to get to see if this tool works or not. So for our new next step, we're thinking to do the same exact tool for other kinases in the same pathway. This will grant us to know the specific function of those kinases and what happens when deactivate one, or when we actually deactivate two. So, lastly, I want to thank my PI, Max Wilson, and Ryan Lack, my mentor, for allowing me to have this cool project. And Gorman, it's been lovely having this summer working full time. It feels great to be a mini grad student. I'm still not grad student. And lastly, I want to thank my whole lab team. They're all very supportive. It's just like a family. Any day that the experiment doesn't work, we just support each other. We look at the future. Future's always looking bright. And thank you. I'm out.